The biomolecules of life are quite distinct from other kinds of chemical compounds. They are organic and use a lot of carbon atoms. They often have complicated structures, emergent properties such as color or toxicity, and they are created only by living organisms. Because they are so distinct from other kinds of chemicals, their structures can seem crazy and accidental, and it can be challenging to find any meaning in their structural diagrams. How are these chemicals made, and how can we understand them? Firstly, it is important to understand that the chemicals of life are not made by adding carbon atom to carbon atom until the end chemical is made. Rather, smaller building blocks, each with multiple carbon atoms in them, are pieced together to make the larger compounds we can find. The larger structures can be taken apart into simpler ones. We will understand the simpler units to understand the large structures. There are 4 kinds of single unit compound, and they each have their own tree of distinct structures. The single unit compounds are glucose, the acyl group, terpene units, and amino acids. They are the monosaccharides. Monosaccharide means single sugar. They have a distinct structure. One popular example of a monosaccharide is glucose. The name monosaccharides describes them very well, as all of the more complex sugars are made of these simpler single unit parts. The monosaccharides have many OH groups in their structures, and this makes them highly soluble in water. These hydroxyl groups are oriented differently in each monosaccharide. This changes the structure of the sugar and gives rise to similar, but very much distinct compounds, of which there are many. Acyl groups are metabolic intermediates. They are high in energy, but they lead to their own tree of distinct compounds. They are transferred between different reactions in the cell perched atop a coenzyme A protein, which helps stabilize it and helps other enzymes recognize the acyl group. CoA is a protein that has an adenine unit, a sugar unit, and a protein unit in it, which all participate to give it its special properties. The terpene units. The terpene units are DMAPP and IPP, which are interconvertible. They are high energy intermediates that are not really useful to organisms on their own, but they are used to build more complicated, very useful compounds. The hydrolysis of the high energy pyrophosphate bond, which is almost identical to the high energy ATP pyrophosphate bond, drives their use, just as ATP can drive other reactions. In this way, they have their own battery pack, and don't need ATP for their incorporation into larger molecules. Amino acids are best known for making proteins, and that is in many ways their primary purpose, but they can be used to make other molecules as well. There are about 20 main amino acids, and they have many different structures and features. They have an amino group and a carboxylic acid group that gives them their name. These 4 groups of single unit compounds each lead to their own distinct tree of multiple unit compounds, but they themselves are highly interconvertible and are made from each other quite easily. There are many pathways that operate between them that are much more complicated than simple trees. Let us look at the multi-unit compounds made from monosaccharides, the polysaccharides. These are usually simple polymers of the monosaccharides. If they are made of just one type of monosaccharide, then they are called homopolysaccharides. But if they are made of multiple different kinds of monosaccharides, then they are called heteropolysaccharides. They are chains of monosaccharides that still have the many hydroxyl groups. They can be categorized by the number of monosaccharide units, 1, 2, 3, some, or many, and by the type of monosaccharide used in making it. For example, a glucan or a fructan. They can be very strong if the chain length is long, which is exemplified by cellulose. The properties of these polysaccharides are usually somewhere in between the high solubility but low strength of glucose or table sugar, and the high strength but insolubility of the fibrous cellulose. This means that they can be used to provide strength and solidity to an organism, like the wood of trees. Polysaccharides, because they are made of small sugar units, are also used as a convenient way to store energy in a condensed form for when it is needed more. Examples of this being done are starch, glycogen, and inulin. Let us move on to the next tree of compounds, those made of the acyl units. The most common type of metabolite that can be made with acetyl groups is the fatty acids. They are called that because they are found in fats as well as oils. They are structurally quite simple hydrocarbon chains with a carboxylic acid group on one end of the straight chain. The chain length is variable, but usually between 12 and 18 carbon atoms long, and are not branched. They are classed according to their chain length and may have unsaturated bonds at some point in the chain. If they do, they are called unsaturated fatty acids. Some important fatty acids are linolenic acid and palmitic acid. They are found in oil from linen seeds and palm oil, respectively. Because of their long hydrocarbon chains, they are hydrophobic, and the molecules like to clump together, and they are immiscible with water. These are powerful properties which result in the formation of lipid bilayers. The cuticle of plants, which forms a waterproof barrier on leaves, is also derived from these fatty acids. Because they are very energy dense, they are also used as energy storage molecules, and are found in adipose tissue and seeds. Those were the fatty acids. Now, let us take a look at some other compounds made from acetyl groups. The base structure of a flavonoid is tricyclic, with some bonds with oxygen and OH groups that are found in all of them. They are found in plants. They are also subdivided into a couple groups that look structurally similar, but the differences are easily seen if you know where to look. They have a lot of variation in structure that is generated after the creation of the main skeleton. An important group of flavonoids is the anthocyanidins. They are water-soluble and colored in tones of yellow. They are important antioxidants and have many antibacterial properties, but when they are glycosidated, they have colors in tones of blue and purple. They change color depending on whether they are in acidic or basic solution. These properties have given flavonoids a place in flower petals and fruits, such as blueberries and grapes and coffee beans, where they have a dual purpose in providing color and helping preserve the fruit. They are localized in the acidic medium of the vacuoles of plant cells, where their stronger colors show best. There is one last group of compounds made of acetyl groups. Another group, polyketides, are likely the most diverse group of the molecules made of acetyl groups. They have very different structures, but there are some noticeable features that are conserved between them due to their biosyntheses starting out similarly. They generally have a linear carbon skeleton that has every second carbon atom bonded to oxygen in some form, or else with unsaturations. However, this is often not the case. Some polyketides have triphenolic benzene functionalities. If they are in the form of longer chains, they may often be linked into a loop. However, this group is very diverse, and it truly may not be possible for you to tell that the compound is a polyketide from the first glance. These compounds are quite rare and specific to certain organisms, which means you should rarely stumble upon them accidentally. They generally have biological effects, and most are very poisonous. This makes them powerful weapons for any organisms for attacking or defending from something, like usnic acid made by lichen, or the macrolide compounds made by certain bacteria. Those are all the main groups of compounds made from acetyl groups. The next group, terpenes, is likely the largest group of natural compounds, with some estimating at least 25,000 different compounds found in plants. Their structures are many and highly diverse, but they are generally united by the C5 rule. This rule states that all terpenes have their molecular formula with the carbon atoms in multiples of 5. This rule reflects the biosynthesis of the terpenes, with the DMAPP units each having 5 carbon atoms. The terpenes are classified by the number of terpene unit they have in them. There are monoterpenes, sesquiterpenes, diterpenes and triterpenes, as well as some other less common types. They have different properties based on how large and heavy they are. Monoterpenes such as menthol are quite volatile. Sesquiterpenes are still moderately volatile, while diterpenes and triterpenes are not. Their other properties vary with the individual structure of the terpene in question, and since there are near infinitely many, and they are all very different, these properties cannot be determined easily. The biological properties of terpenes are emergent and specific, with some amazing biological properties popping up in random places. Monoterpenes are volatile and are important components of the smells of foods and plants. Sesquiterpenes are also important for smell and taste. Diterpenes no longer contribute to smells, but many have anti-biological properties. In addition, most of the terpenes with some of the most amazing properties are diterpenes. For example, stevioside is a diterpene compound that is a strong sweetener and is found in the stevia plant. Salvinorin, a crazy powerful psychotropic compound, is found in the divine salvia, Salvia divinorum. Taxol, a well-known cancer medication, is derived from a diterpene structure. However, because they are so many and are found in complex mixtures, most of them have not been studied as much as some simple compounds have, so many of their properties remain unknown. Terpenes are found in many different places and have an amazing diversity of functions. Another group that deserves special mention is the steroids. While they are technically terpenoids, they are usually mentioned in a distinct group because of their importance to humans and to life in general. They have a conserved gonane skeletal core, but have some different elaborations on the structure at some points. They differ from each other by these elaborations, and they are best known as the human hormones such as progesterone, testosterone, cortisol and estradiol, as well as cholesterol. They are also able to contribute to the fluidity of the plasma membranes found in all living cells because of their hydrophobicity. Those are the compounds made of terpenoid units. Now let us look at those made of amino acids. Amino acids are very well known for making proteins and polypeptides, and this is their most important function, and I assume that you already know something about that, so I will not talk too much about it, but they can definitely make other kinds of compounds, and we will look at those. A different kind of molecule that can be made from proteins is the porphyrin ring. They are made from the amino acid glycine through a biosynthetic pathway, and their most important function is in hemoglobin. The large ring of porphyrin is made of 4 small pyrrole rings, and it has 4 nitrogen atoms facing inwards. These nitrogen atoms have 2 sigma bonds and 1 pi bond, and a lone electron pair, which faces towards the center of the porphyrin. These lone electron pairs help stabilize the iron atom that lodges in the middle, and they hold it there. The porphyrin ring in hemoglobin has proteins attached to it that help modify and add precision to the oxygen-carrying activity of the central iron atom. Although hemoglobin is the metal-carrying cycle we know best, but there are actually many variations on the structure that have different functions, such as those that carry magnesium in chlorophyll. A different kind of compound that can be made of amino acids is the phenylpropanoids. These are quite distinctive in the feature of them having a phenyl group attached to a propane, that is, a 3-carbon part. These are rarely free molecules, they are usually polymerized as in lignin or as glycosides or esters. Phenylpropanoid molecules are hydrophobic, and the links in lignin heighten this property, making lignin highly impermeable to water. Most also have phenolic groups, and many also have a carboxylic acid group. A significant phenylpropanoid is cinnamaldehyde, a sweet-smelling compound found in the bark of the cinnamon tree. The cross-linked lignin is found in the wood of trees, where it stops the water in the water-conducting organs of the tree from leaking, which is critical for trees being able to grow upwards. Finally, a very complicated and extremely interesting group of compounds is the alkaloids. There are some historical rules for what an alkaloid is. 1. They are found in nature. 2. They act on the central nervous system of vertebrates. 3. They contain a nitrogen atom in the structure that is in a cycle. And 4. They are biosynthetically derived from amino acids. Examining these historical criteria under a modern lens, we see that they are no longer good enough to define what an alkaloid is. While the first and second rules are true, they are also very broad. The concept of an alkaloid as something natural has remained true, and modified or synthetic compounds derived from them are not referred to as true alkaloids. Their activity on vertebrates, the second rule, is also generally true, though it is more of a coincidental one, as the compounds that follow the other rules also tend to be bioactive. For the third rule, even though all alkaloids contain a nitrogen atom, not all have it in a cycle, for example ephedrine. These are sometimes moved into an artificial category, pseudoalkaloids, but this category is only really based on the idea that all alkaloids must have a nitrogen atom in a cycle, which was simply a historical observation at first. The fourth rule, however, no longer gives a valid criterion for what an alkaloid is. Although we are examining the compounds made from amino acids, many alkaloids are made from a terpene structure, but through incorporation of amine groups, such as delphinidine and psilocybin, which is actually based on a steroid carbon skeleton. Despite this, a great many alkaloids are in fact made from amino acids. These include many economically and medicinally important substances used by humans for their effects, such as nicotine in tobacco, morphine and codeine, as well as quinine, used for treatment of malaria. Many psychoactive drugs, especially those that have been used for many centuries, are alkaloids, such as cocaine, caffeine, and the tryptamines, are all alkaloids. Newer synthetic drugs, however, would not be considered alkaloids because they are not found naturally. Alkaloids have a huge diversity of structures and are all quite interesting, so research them yourselves if you want to know some more. Two notable types of compounds that do not exactly fall into any one of the groups are the organic acids and the nucleobases. They are important and have central roles in the chemistry of cells, but they do not exactly have an origin in any one of the 4 groups already mentioned. Rather, they are the products of the combination of multiple groups. The organic acids are quite simple small molecules with carboxylic acid functional groups. They have a chain length of generally no more than 6 carbon atoms, are sometimes branched, and often have alcohol, ketone, or olefin functionalities. These may seem very similar because their distinguishing features are relatively mundane, but their interconversions and reactions are central in the metabolism of all cells, with all those functional groups participating, such as in the TCA cycle. The nucleobases are guanine, adenine, uracil, and thymine, as well as cytosine. They are made from amino acids and the saccharides together, as well as taking parts from other sources in the cell, although their end structure bears no resemblance to their precursors. They are used in DNA and RNA, and some end up being used in secondary metabolism, for example caffeine, which is usually considered an alkaloid. It is important to understand how exactly the great diversity of chemicals occurs. One factor that it is important to recognize is that there are actually relatively few carbon skeletons that can be made for any given size of terpene or polyketide. The huge number of compounds that it is possible to generate comes from the modifications that happen afterwards. These mostly come from variably oxidizing the skeleton, and because this can be done in many different positions for any molecule, there are a near infinite amount of end molecules, each with their own properties. Sure, they can be related in their activity if their structures are similar, but they are also distinct molecules that are not the same. This post-skeletal functionalization generally proceeds through simple steps. There is oxidation, which is done by molecular machinery called cytochromes, that can turn a CH bond into a COH group, and then those can be turned into ketones, or aldehydes, or carboxylic acids. There is dehydration and dehydrogenation, which create double bonds. There is methylation, which exchanges an OH or NH bond for a methyl group. And then there is esterification. Esterification usually proceeds with the addition of an acyl group onto an alcohol group, or onto an amine group, but any carboxylic acid can do it. This includes the acids of phenylpropanoic acid type compounds, like cinnamic acid. Glycosidation can also occur, which connects a saccharide to an alcohol or other group. Many structures, however, are a mixture of groups within their core. The synthesis of catharanthine shows how amino acid-derived compounds can be combined with terpenyl groups to create very complex structures. Tetrahydrocannabinolic acid is also a good example of this, where a terpene group is united with some other kind of group to give a synthesis of both. Investigating these natural products is very difficult. For one thing, these compounds exist in highly complex mixtures in organisms. Investigating and researching a single chemical requires separation and purification of that one chemical, which becomes extremely difficult when there are many different compounds mixed together, especially because the chemicals can be low in concentration in the starting material and sensitive to the purification methods. Even if the chemical is purified, analyzing it and determining its structure is tremendously difficult and requires a lot of experience, patience, and equipment.